It's time for our next speaker of the evening. May I present to you Andrew Hessel. Andrew is joining us live from San Francisco. Welcome, Andrew.
Hi.
How are you?
I'm doing pretty well this morning. Thank you.
That's great. We just finished our conversation with Dr. Akash Goyal from New York, and now we're switching coasts from the comfort of our couch here in India. Quickly to introduce you, Andrew. Andrew Hessel is the president of Humane Genomics, a seed stage company developing synthetic viruses, starting with virus-based therapies for canine cancers. He is a co-founder and chairman of the Genome Project-write, the large genomes, including the human genome. It's an international scientific effort working to engineer these large genomes. From 2012 to 2017, Andrew was the distinguished researcher at Autodesk Life Sciences. If you have some time, check it out. It seems like a sci-fi film. He has been Singularity faculty since 2009. His goal is to help people better understand and use living systems to meet the needs of society. Welcome to FutureBets, Andrew.
It's great to be here. Thanks for having me.
I did see some of your videos, and I think my background in biotech helped a little bit. However, they were very well articulated and explained. I'll be your moderator for the evening, Andrew. The format would be that you would speak for about 20-25 minutes or so, and it'll be followed by Q&A moderated by me. Quickly, my background is I have a master's in biotech enterprise from Johns Hopkins, and I have a biochemistry degree. I spent some time at a healthcare-focused investment bank on Wall Street. As you know, Ashika Group, we are a full-fledged diverse financial services firm. We're 25 years old. We do all your brokerage, investment banking, lending, wealth management, and asset management. Great, so I think we can just get started. Over to you, Andrew.
Terrific. Well, I'm not going to use a slide presentation today, but I'm going to give an overview of what I've been seeing and working on really for a significant fraction of my life. You just froze. Are you okay?
I am, yes.
Okay,
I'm just
great.
you the floor. Yeah.
Yeah, and also I just sat in on the last presentation. Thank you so much for doing that. We are truly living in unprecedented times, and I don't know. I'm actually based out of New York, but I was really concerned with what I'd been seeing in the last few weeks. So we relocated to the West Coast where we have another property. Anyway, my main interest in life really is how does life work? And living systems are the most complex machines we know about, but very few of us spend our life paying attention to really studying it. Only a fraction of people are life scientists, and of course many people that are interested in life go into medicine and don't study the fundamentals. So when I had to start making a decision about what I was going to do with life, because I'm interested in all technologies, I started to focus in on cell biology, really looking at the smallest individual unit of cells, of life, and how the mechanics of how the cell operated, but also how it was programmed. So moving into genetics. Today, I've come to realize that just in general, people don't think about life because so many of us have moved to urban settings over the last 100 years that we're getting more and more divorced from the life that's around us. You can see I'm living in a redwood forest here, so I'm constantly surrounded by living things, but it's a completely different situation in New York, where really the only organisms that are around us are ornamental, the plants and parks that we have, the invisible, the microbes, and now the viruses that are disrupting us. There's the organisms that we tend to bring into our lives and domesticate, like cats and dogs. Dogs are everywhere in New York. there's the raccoons and other pests that have learned to coexist with us in the cities. And then there's just, you know, things that we just want to get rid of, the spiders and cockroaches and other things, but we don't think about life. And even the basic unit of a cell, which is again the most complex machine that we know about, is usually, our understanding of it, our visualization of it is typically quite cartoonish. But I've been fascinated by this. So I usually lead people into, you know, the types of things that I'm interested in by just focusing on the simplest cells and giving people a mini background to them. And the simplest cells that most of the scientists study are bacteria. You know, most people have heard of E. coli. E. coli is an organism that's existed on this planet for almost 4 billion years. It's an elegant little cell that is kind of the wise old man. And so many researchers study it. It's the main component of one of the largest by number bacteria in our guts. And so we've co-evolved with it. It helps us digest our food. But it's an amazing little organism with a genome of about 4.5 million base pairs of genetic code. So the types of things that I see happening and has been a part of my whole career is that we're getting better tools to look into biology, molecular biology. Think of them as microscopes. Of course, we've had light microscopes for a long time, but the resolution of a light microscope is quite low. It's about 1,500 times. We've developed electron microscopes that give us a much higher magnification, but you can't look at living tissue with an electron microscope. So you're always looking at an artifact. But more recently, we've had technologies like cryo-electron microscopy, which takes electron microscopy, the images, and computationally assembles them in the same way that we can make very elegant computational assemblies of radio telescope images. There was a really fascinating technique that came out a couple of years ago that was called a DNA microscope. And it's a way of tagging molecules in the same way and tracking them in a cell the same way we can track a cell phone today in the world. And so it gives you this roadmap of molecular interactions that's really quite beautiful. And there's more and more of these microscopies coming online. And in fact, many of them have won significant recognition in science, including Nobel Prizes. But so now we can actually see more and more into this nanoscale world of the cell, because the cell itself is fairly large. It tends to be micron scale. You still need a microscope to look at it, but the components inside are nanoscale. And now that we have a better way to look at them and look at these systems and to keep track of, and even to go into that world through various visualization and digitization technologies, it's the cell, the inner guts of the cell is becoming a lot more real. And then of course, DNA technologies have really advanced remarkably over the last 40 years. It's humbling to recognize that we only cracked the genetic code in the late 1960s, early 1970s, and started to do this type of manipulation. But in general, DNA is just a language. We talk about our genome as being the book of life. Well, DNA is the language, and it is recognized more and more as a programming language. But like any language, you have reading. You have to be able to read the language or code. You have analysis. You have to be able to comprehend the language. And then you have writing. Reading is always where we start to learn a language, any new language. And we've gotten remarkably good at reading DNA code. even when I got started in life science, reading DNA code was really slow and technically challenging, very inefficient. But as automation has come to the DNA sequencing world, and a lot of technology development has been supported in large part by the creation of the Human Genome Project in the 1980s. DNA reading has become very high throughput, very inexpensive. And I think the current cheapest price for a whole human genome is about $300 with the data plan, which is remarkable considering, you know, to generate the first human genome was an international effort that took 13 years and cost $3 billion or more. There's even though we've hit remarkable price points, we still haven't learned to leverage DNA reading like the technology of our phones. Very few people wake up and go, I'm going to get my genome completely sequenced and analyzed today. But maybe we'll hit that point in the next decade or so. We're already seeing new technologies coming online that could take DNA sequencing of our genome or any living thing down to, but just using our genome as a baseline, well below $100, and soon I believe it'll be cheaper, the cheapest diagnostic test that a doctor can do as we walk in the front door of their offices. We're actually seeing the emergence of a field called molecular electronics, where the very robust electronic tools that we have today that power the world can directly connect to biological systems and gives us a level of sensitivity that we've just never had before. Of course, we know that DNA is not easy to read. The computer-based tools for analysis and comprehension of DNA have gotten remarkable. Whole digital pipelines that can analyze complex genomes in a very short period of time. So overall, the world of genomics has been exploding. And we've seen this even with the current COVID-19 outbreak. The virus that is responsible for this outbreak was sequenced in less than 2 weeks and put online once it was recognized as the source of the outbreak. And this is amazing because with the SARS outbreak, 15 years earlier, 15 or 16 years earlier, it took over a year just to sequence that virus and get that information to researchers around the world. The thing that I'm seeing, though, that's the real game changer, and that has been an increasing part of my life, is that we're learning how to write genetic code now. Like, reading is taking the molecule of DNA and digitizing it. Now we're learning how to go the other way, take digital information and turn it into the molecule of DNA. And effectively, this allows us to start programming the cell. This technology is based on a chemistry that has been around for a long time called phosphoramidite chemistry. The chemistry has a limitation that it can only write short fragments of DNA. So you have to do a complicated assembly process to make long chains of DNA that you would need for a whole genome. But we've gotten remarkably good at doing that over the last decade. So I've seen whereas 20 years ago it would be a challenge to write a few thousand base pairs of genetic code sufficient to make a single protein. We have quickly advanced to being able to write 50,000 or 60,000 base pairs of DNA code today pretty much effortlessly, which is powering a whole field of cell programming called metabolic engineering. And at the extreme end, where people are starting to really focus on how do we do bigger DNA synthesis and assemblies, we're we're starting to push into routine million base pair constructs, which is getting to be the size of small bacterial genomes. And last year at the Genome Project-write meeting, we had two presentations that really showed how far we're going. Jason Chin from MRC in the UK talked about the assembly of the E. coli genome, which is about, the one that they assembled was about 4 million base pairs of code. This is absolutely remarkable. And then an international team of scientists that have been working on the yeast genome, yeast being the foundation for bread and beer, and brewer's yeast, announced that they'd finished 99% of the synthesis of that genome, and now that they were in the final finishing phase. So moving forward, we're now entering a period where at the leading edge of genomics in writing genetic code, which is unlocking the power of the cell and biology, we're now getting to the point where we can write entire chromosomes, whole genomes of smaller organisms, but entire chromosomes of larger organisms like plants, animals, like mice and other research animals, and ourselves, potentially. So this is, we're starting to really get into some challenging areas of not only science and technology development, pushing reading and writing of DNA to new levels, pushing design tools for this type of work to new levels, but also reaching some really fascinating questions that we have to start asking ourselves as a species as to, you know, what the ethical issues and intentions that we want to create as we start really mastering the fundamental technology of a living world, and looking at where do we want to see ourselves go as a species. So I could continue, but this is really the heart of it. We are now seeing the emergence of a genetic programming industry that is not so different than the industry that creates hardware and software for our computer networks. Over the last my lifetime, I'm 56, the world has completely digitized. So many of our systems now are dependent on digital systems, and thankfully they connect us in ways like this. The planet's become a lot smaller, but now we're learning to tap into a completely different network architecture, which is the network that nature created, and now we're learning how to program it. We're not good at it yet. Our tools are really crude. Our processes are slow, but it's pretty clear that it's accessible to a larger community, just as computers are, and that it will be one of the fundamental foundations and probably economic drivers of this century. And it's a technology like computers that just won't stop as we continue to develop it. I'll leave it at that.
Thank you, Andrew. Thank you. There's so much to take away from that talk of yours. I knew cost of genome sequencing had defied Moore's law. What I didn't know was that it could be the cheapest diagnostic in the coming future. That is somewhat of a revelation. Andrew, you compared a cell to a computer. You called DNA the programming language. So it's essentially biology meets computer science. Two things that Indians are famous for all over the world and at home. What will it take? I think this is my first question, my first obvious question to you. What will it take for India to start innovating in these areas? Since, you know, we have the resources, how can we leverage our existing pharma expertise and resources to achieve more in healthcare? And what would be the tech background required to bring these technologies to the country?
That's a great question. And it's been 20 years since I've been on the ground in India. So it's changed a lot. But really you've got all of the necessary pieces to be a powerhouse in genomics. Again, you've got incredible education in engineering and science and technology. You've got computer science as a major foundation. And I want to say, as you start moving into synthetic biology, using DNA as a programming language or analyzing DNA code, you're not wearing a lab coat so much. You're sitting in front of a computer. You need the computer skills. It's the foundation. And so I don't have a great sense of the pharma industry in India, but in general, the pharma industry is based on an older time. Most of it is still the production of various chemicals. The idea is still mass market chemicals rather than custom precision biomolecules for a particular disease. But I think that as these technologies continue to be refined, become cheaper and more accessible, I believe India is going to just be one of the most dynamic areas in the world for biotech. Not to mention all of biology is powered by sugar or sunlight, essentially, at the lowest levels. India is rich in the resources necessary for life.
That's great to know. Didn't know you were also collaborating in India. So now you know who's your next collaborator going to be. It is going to be us at Ashika. Going forward, Andrew, I would love for you to touch up on CRISPR. There are already a bunch of companies that have IPO'd in the US around CRISPR. You have Intellia, you have CRISPR Therapeutics, you have Editas and whatnot, with some even touching the $1 billion market cap. Can you talk about where they are in terms of becoming successful at disease management, and how do you see the regulatory landscape shaping up for these companies?
So CRISPR is an amazing technology. It's actually part of a bacterial immune system protecting it from viruses. So viruses have always been the biggest agents of evolution and education in life science. We're still experiencing that. But CRISPR technology allows you to make precise changes in a large genome. And our genomes are very large, like 6 billion base pairs duplex. So it's exploded on the scene as a very useful technology for manipulating genomes. And as you pointed out, there's a number of companies that have already started to apply this to, in general, human therapeutics, because we can't synthesize a human genome today. Like, that's beyond our current capacity. That's actually the heart of the Genome Project-write. It's just as in 1990, the Human Genome Project was launched to read the human genome. The Genome Project-write goal is to be able to write a human genome, not to make babies, but just to be able to do that complex engineering. So right now, CRISPR is the only way we have to manipulate a plant or animal genome in a precise way, very inexpensively, very cheaply. That's why it's exploded, and these companies are quite powerful. And of course, the technology is still evolving. We're learning how to apply different Cas enzymes that do different things. I think the technology is going to continue to explode and be used therapeutically, because we can do precise changes even to an individual with CRISPR. So it's going to be a very powerful tool in doing things like correcting genetic disease. We've already seen it applied to human embryos. It's essentially genetic surgery. If an embryo is damaged, you can make a precise change. Of course, it also opens up the ethical issues that you can make an enhancement to the human genome or to a plant genome or other genome. So that's opening up whole new areas of ethical discussion that we're going to have to solve fairly quickly. But it dovetails with the type of work that I'm doing with synthesis. I don't use CRISPR for the work that I'm doing with virus engineering, because I can make all the edits in the virus genome just using computer-based tools, and then hit print and synthesize the full virus genome. So CRISPR isn't very useful for editing a virus today. In the next few years, CRISPR won't be useful to edit a bacterial genome, because it'll be easier and more efficient to just synthesize the full microbial genome. And eventually, and this is looking a little further down the road, we will be using synthetic-based technologies to replace CRISPR even in large genome engineering.
Interesting. I'm glad you mentioned the designer babies. Jennifer Doudna, one of the pioneers of CRISPR, requested a global pause on human embryo editing. And yet you have the scientist in China who performed these experiments and led to the birth of 2 girls. Absolutely, there are ethical aspects to be considered, but where do you think the regulator, the FDA's narrative is going to go towards?
Well, I think it's tricky because the case use for engineering humans is one very hard to regulate. Let's just take a look at. I have a 2-year-old and a 5-year-old. Both of them were made using IVF technologies. In some sense, they're already engineered humans. They exist because of work that has been done since the 1970s to develop and refine the in vitro fertilization process. And I think that in some cases, again, when you're dealing with parents, it's an N of 1 relationship between the scientists and the, let's just say, the application, in this case, making a human life. I think that's really hard to regulate. Even IVF clinics are very difficult to regulate. So I think we're going to see anecdotal use of these technologies, just no matter what the regulatory environment is in one particular country. Remember, there's almost 200 countries, right? So there's no rationalized scientific framework or regulatory framework for life science. The big issue a couple of years ago with the CRISPR babies was that it wasn't, in my opinion, they missed the mark in the sense that it was an enhancement. It was not medically necessary to make these babies resistant to HIV. Going back to IVF technologies in the 70s, the reason why this was accepted was it brought children into the world that would not have been born otherwise because of medical issues. So there, even though it was sensational at the time, it was very quickly accepted because it became a powerful assistance for infertile couples. And I think if they had used CRISPR in that way out of the gate, to use the technology to repair a defect in the embryo that prevented it from fully developing and becoming a baby, I think it would have been celebrated and not reviled.
Wow, that's a very unique perspective on that. But definitely is going to be, like you mentioned, a regulatory challenge. It's going to be a massive regulatory hurdle.
I think it's really hard to regulate life because it's so universal. Mm-hmm. It's one of the challenges that we talk about as we develop a synthetic biology. How do we create a regulatory foundation that's universal? How do we share best practices? How do we instill the right culture of doing good? How do we create an ethical foundation that is universal again across all cultures? That's really difficult, and I don't know how to do that. What I know we need to do, and I think that this current outbreak is giving us a tremendous education in, is I'm not so worried about the application of synthetic technologies to microbes or plants, animals, even to us. I think the biggest thing that we need to be concerned about is that as it gets more powerful, and gets more democratized, which in general is a good thing. If we just look at computer technologies, it increases the possibility that someone will do something either in error or by intention to make a virus that could be harmful, either a pandemic or a more targeted bioweapon. And I've been speaking for years that we have to create a new foundation for virology as our tools for engineering viruses becomes more powerful. And I've seen it become more powerful. I made my first synthetic virus in 2014 while I was working at Autodesk, not a biotech company, a software company. And today I have a company based in New York that does viruses, that engineers viruses, and our cycle time to be able to make a virus, design, build, test, is down to something like 2 weeks. And that will still accelerate. So we need, this outbreak is giving us an opportunity to recognize the power of biology over us. It's giving us an opportunity to understand that yes, this is a virus that appears to have been selected in nature through a series of events that is now giving us a global lesson and unifying us in some ways. But it couldn't have come at a better time, since we are also now able to design and synthesize any virus from scratch. And that comes with some real risks. So now we get to build essentially as a species, as a global community, a new foundation for biosecurity. And if we do not do that in the as a follow-on to coming out of this outbreak, we will have failed. Something really significant. This is a lesson we need to learn now.
What do you think is going to initiate this process of biosecurity?
I think that I think it's COVID-19, because we haven't had a pandemic of this type for over 100 years. So the knowledge that we got out of the 1918 influenza has been lost. It's just the lessons learned have died off over the century. Of course, the world is completely different than in 1918. The dynamics that produced the pandemic are completely different. But you know, the lesson, so we're learning a new lesson about how vulnerable we can be. And look, it's not like we haven't invested over time in basic virology and sensing and having pandemic responses, etc. It's just that we've learned that they're not very robust. A lot of it has, it's like you put a lock on the front door and you realized it was easy to, to break the front door down. So it's like, so I think this is just a real experience to stop and in the aftermath, think about how we build effective global biosecurity for infectious agents. And we've got the tools and technologies to do that. And now that this is a multi-trillion-dollar outbreak that has touched individuals by having to shelter at home or losing their job or just not being able to go to work, I think now we'll see a tremendous investment in preventing this type of disruption from ever happening again. And this is the first incentive that we've had of this type in a century. So yeah, it's a big deal.
To elaborate slightly on that, in what scenario are you seeing in the US such preventative measures being taken, A, from a healthcare system perspective, and B, from an individual perspective?
I've been really, to be frank, I've been disappointed with the U.S. response. First of all, and it's been discussed so widely that this particular administration has largely torn away a lot of this, stripped away the pandemic surveillance. Clearly, many of the institutions that we've relied on to support public health have been eroded. And in general, I'm Canadian living in the U.S. I've been down here since 2012, since I joined Autodesk, first on a visa and now I'm a permanent resident moving towards citizenship. This is the craziest healthcare system ever. Like, no country in the world says, I'm going to go model my healthcare system on the U.S. healthcare system. because it's incredibly expensive, fragmented, and with poor outcomes. So I think this is a real opportunity to recognize that we need some level of public health, universal health, that spans the globe. They has to work together quickly, whether it's China and the US or India or any other nation, has to link together. It has to provide a basic level of detection and sensitivity and treatment, if only for infectious disease. Because that infectious disease doesn't care about borders or politics or how rich you are or anything like that. We're its host. That's it. It's just opportunistic. So I think this is an opportunity to rationalize and modernize the U.S. healthcare system and every other healthcare system in the world, because we're learning what works and what doesn't, kind of on a scoreboard of countries right now with this particular virus. So hopefully we'll share best practices and make investments in a new foundation moving forward.
I've had my fair share of interactions with the US healthcare system while I was living there. Okay, moving on. Andrew, we've had biologics, we've had gene therapy, we've had personalized medicine, we've had RT-TCR therapies and whatnot. What is the next big innovative thing that you may have heard in your biotech circles?
Oh, I'm an evangelist for DNA writing technologies. First of all, DNA sequencing. DNA sequencing, reading DNA, as I said, is likely to become the most powerful diagnostic for, the fact that we can get our whole genome, not just sampled, but sequenced today for $300 or thereabouts is insane. In terms of the amount of data, high value data that we can generate about ourselves for that price. I'm on record as saying once you kind of hit that price point, there's the opportunity for a company to come along and just sequence you or me for free, because they can learn so much about us and amortized over a decade, essentially introduce us to products and services that will more than recoup that investment. A customer acquisition charge, so to speak. And there's a company, Nebula, that is very close to being able to do that for everyone. They're leading the charge on this low cost genome sequencing and analysis in a way that I find to be really good. So I've been advising them for the last couple of years. But it's genome writing that is the game changer, because already it's powerful enough to start working on microbes. It's amazing when you can, you see 10-year-old kids today, even younger, sometimes sitting down in front of a computer and writing powerful software. In fact, one of the largest COVID tracking sites was written by a 17-year-old. Which is, again, amazing. Facebook, we forget, was started by like a 19-year-old freshman. Now we're starting to see that type of capability and energy come into programming biology. Because you can, with the right tools and processes and support today, it's not as efficient as I would like, but you can teach a 10-year-old to program a cell to do something useful. And by the time that person, given the pace of change of the technologies, by that time that 10-year-old is 20, they can do things that would make today's biotech companies look pretty slow and pokey. So DNA writing technology, as it advances, and it will advance along pretty much the same price performance curve as sequencing, just lagging about a decade, is going to just change the world of biotech. And I don't think people appreciate how much it will change the world of biotech, but it basically makes biotechnology, genetic engineering accessible to just about anyone that wants to invest the time and effort. It will get more seamless in terms of the design, build, test process, more automated. You won't need a lab in your home. There's already cloud laboratories that you can go and program. And I think that we'll see kind of a Cambrian explosion of young, smart, highly automated, highly digital biotech companies in the future that will give today's industry some real serious competition.
That's excellent. So I know you don't, you're not focusing on the commercial applications, but just I mean, it's a fair next question. What are the commercial applications? Where do you see biotech companies propping up to solve what problems, what unmet medical needs can be solved by this?
Well, there's so many of them. It's like asking, what can computers do? You know, it's like we're surrounded by life and we're surrounded by problems. Look, the beautiful thing about life is it's the most powerful and efficient manufacturing process. So, like, even if you look at everything we make from plastic today, plastic is just biological goop that companies drill into the ground, suck it up, and refine. With biotech, you don't have to drill into the ground to get biogoop. There's plenty of biology around us. You can suck carbon dioxide out of the air if you need structural material, the carbon. And you don't need to refine it because you're making the exact molecules you want in the first place. So you don't need the big refineries. So it's just a clean, powerful industry. So just making plastics and structural materials becomes, just much more advanced, let alone more complex machines, because the cell is like a 3D printer, but it makes tens of thousands, even hundreds of thousands of different compounds, but it's a programmable 3D printer. And it's just like, that's what's being unlocked here. So basically, we're going to see life technologies reach into every area of human activity and make it smarter and cheaper and more efficient, because it is the most powerful technology. We're going to see more diseases cured. We're going to see new plants and animals created. We're going to see new materials made for manufacturing processes. We're going to see us clean up the world, because we don't have to bulldoze a forest to put up a chemical manufacturing plant to go and make biology. We can just reprogram the life in that region and put it in, essentially a bioreactor. I'm also a big fan of leaving nature heal and moving these bioreactors for making materials or drugs or things we can't imagine today, moving them into closed environments, putting them essentially underground. Because biology, engineered biology doesn't need to be on the surface. It doesn't need sunlight. We can feed it the raw materials it needs to grow. So you can do this in dark factories. And you can have those dark factories anywhere. So you might as well put them in places where there's a physical separation from the natural world, which we don't understand because we've looked at the natural world as a resource instead of a garden, that we should be, learning about and cultivating.
Interesting. I think, Andrew, conversation between you and I can go on forever, but I've been signaled that we have some time issues. So just before I let you go, I think you've talked about this intensively, but I have to ask, we ask all our FutureBet speakers the same question, this one last question. What is Andrew Hessel's FutureBet?
So my FutureBet is that humanity thrives, but I think that we're going to move off the surface of the planet. My FutureBet is that the planet will go back to a natural state on the surface, our oceans, our land masses, and that humans will start to move underground into essentially defined environments, closed environments where we can be much more sustainable. And the lessons that we'll learn as we go down will help us move into space, because everything we put into space, whether it's in orbit or moon bases or Mars, and we love that type of exploration, they're closed systems. So you don't build closed systems. You don't want to learn about doing closed systems when you're putting them on the moon. Imagine you learn about them here on Earth. And right now, part of the challenge we're seeing with the pandemic is we don't have a closed system. Our world is churning all these microbes and et cetera. I think as we start to build more more livable closed systems, more sustainable closed systems, I think humanity just will not only reach into the stars, but be more secure and have abundance that we just, you know, we take for granted today.
Wonderful, Andrew, wonderful. Your future bet sounds like another plot of a sci-fi film. I love it. And to all our listeners, if designer babies interest you, I would recommend the movie Gattaca. I think it's the best sci-fi film out there in terms of gene editing. Okay, that's fantastic. I think that is our time, Andrew. Thank you so much from the entire team of Ashika for giving us your precious time. And if we have any further questions or our participants have some questions, I'll send them to you.
Terrific. Thank you so much.
Thank you, Andrew. Take care.
Okay, bye.
Bye.